Hello, hi, namaskaram. My name is Kirtan and I am a second year medical student. Welcome to my channel Physiology at Once. Today I will be talking about the topic salivary glands and their secretion. Similarly, I will be talking about the secretion of the main glands like gastric or small intestine secretion in the upcoming videos. So, if you haven't subscribed to my channel, please subscribe because it's free and also you can get the notifications for these types of videos. So, let's start. So today's topic is salivary glands and their secretion. So in this video, we'll talk about the salivary glands, the composition of the saliva, the function of the saliva, the mechanism of the secretion of the saliva, the control of salivary secretion, and some applied physiology. So let's begin this video. So salivary glands, they can again be divided into major and minor salivary glands. In the major salivary gland, they contain parotid, sublingual and submandibular gland. So there is one pair of parotid, sublingual and submandibular gland. And they can also be divided again based on the nature of secretion, like the mucus secretion glands, the serous secretion glands and the mixed secretion glands. The mucus secretion glands is nothing but the glands or the salivary glands which release the secretion which is thick in consistency and contain mucus in it. The serous type of salivary gland contains the secretion which is watery in secretion, and the mixed type of salivary gland contains or releases the secretion which contains both mucus and watery like secretion. So let's talk about three of these glands detailedly. The parotid gland, this is the largest salivary gland and it is entirely serous gland. So it is a serous gland and does not produce mucus secretion. This secretion is poured through the parotid or Stenson duct into the mouth. The next one is the sublingual gland. It is the predominantly mucus gland. Its secretion is drained into the mouth through the sublingual ducts, or it's also called as ducts of Ravenous. The next gland is submandibular gland. Its secretion is drained to the submandibular ducts or the ducts of Wharton. It is the mixed type of gland, serous and mucous type of secretion. So you will study about the salivary glands in the anatomy in the head and neck unit. So I am not detailedly discussing about the salivary glands in this video. Instead we will focus on the salivary secretion and the composition of the saliva. So composition of saliva. Saliva mainly contains two components, that is water and solid. Water contains the major portion which is 99.5%. And solids are only 0.5%. These solids can again be divided into organic and inorganic. Under the organic, it contains enzymes like ptyalin or salivary amylase, lysozyme, carbonic anhydrase, RNase, DNase, or etc. Under the inorganic, they contain cations like sodium, calcium, potassium, and anions like chloride, bicarbonate, phosphate. Saliva is actually hypotonic to the plasma because it contains less concentration of the sodium and chloride than the plasma. It also depends on the rate of secretion. The tonicity of the saliva depends on the rate of secretion. Like when the rate of secretion increases, or when the saliva is secreted faster in the salivary glands and flows through the duct, its tonicity increases than the plasma. The pH of the saliva is alkaline, that is about 8 pH. So actually when it is released from the acinus or the glandular cells, it is actually acidic. But when it flows through the duct of the salivary gland, it becomes basic or alkaline due to the increased production of HCO3. Increased production of HCO3 makes the saliva alkaline, which I will be talking about it now in the mechanism of secretion. So now let's talk about the functions of saliva. Why should we study about saliva? Like, why is it important? So the main function of the saliva is the reduction of the starch to oligosaccharide or splitting of starch. It is due to the presence of ptyalin or salivary amylase. So it cuts down 30% of the carbohydrate, or it helps in the digestion of 30% of the carbohydrate. And in the deficiency of the saliva also, the digestion does not stop. because there is gastric secretion which helps in the digestion of the starch. The next function is the protective functions. It prevents the oral infection. It also contains lysozyme, which is antibacterial. It contains immunoglobulin A, which helps in the local immunity process. It also contains lactoferrin, which is again a bacteriostatic enzyme. The third function is that it keeps the mouth moist, which facilitates speech. So you might have observed when the mouth becomes dry, it will be difficult for the speech. It increases the taste of food. So the taste buds which are present on the tongue receives the taste sensation when the food is dissolved properly. It is helped by the function of the saliva. It helps in the mastication and the swallowing process. Mastication is nothing but the chewing process. And without the dissolving of the food materials with the saliva, it will be difficult for the swallowing process. So the dry foods are difficult to swallow. It also acts as buffers to the gastric acid to some extent and prevents heartburn. It excretes certain heavy metals, thiocyanate and morphine from the body. So helps in excreting these toxic materials. Now let's talk about the control of salivary secretion. Before that, we'll talk about the mechanism of the secretion of the saliva. So it occurs in two stages, that is secretion in the acinus or the glandular cells and the secretion in the duct of the salivary gland. The secretion in the acinus, it is rich in amylase or the ptyalin. The electrolytes secreted from the epithelial cells are sodium, potassium, HCO3- and chloride ion. The fluid of primary secretion is almost isotonic to the plasma and is acidic in nature. So what happens in this is the acinus cells or the glandular cells contains granules which contain the amylase enzyme. So when there is stimulation of the salivary gland, these granules are exorcised from the acinus cells and the amylase enzyme is released. So next, let's talk about the next stage, that is secretion in ducts. This secretion which passes through the intercalated and striated ducts are modified. So therefore it's called modified or final secretion. How is it modified? The Na and Cl are extracted from the fluid and the K plus and HCO3- are added to the fluid. So therefore it becomes alkaline while passing through the ducts, which I talked earlier now. So this, let's understand through the diagram. This is the acinus cells or the glandular cells which release K plus, Na plus, Cl minus, HCO3- and enzymes, and this is the primary secretion. So while coming down in the duct, the Na plus and Cl minus is abstracted and HCO3- and K plus is added to the secretion. So therefore it's the modified secretion and the pH of the saliva becomes alkaline in this stage. So now let's talk about the control of the salivary secretion. How is it controlled? It is controlled by the neural mechanisms, that is parasympathetic and sympathetic. Let's talk about the parasympathetic stimulation now. It mainly occurs by chewing, sight, smell and taste of the food. So these are the main stimulants for the parasympathetic stimulation. It increases the salivary secretion rich in enzymes and mucin. It also increases secretion by vasodilation and stimulation of the glandular tissue. So vasodilation is important in the parasympathetic stimulation. It acts to the salivary nucleus which is present in the medulla. The next one is the sympathetic stimulation. It temporarily increases the secretion, but finally it decreases it. The sympathetic fibers to the salivary gland originates from the superior cervical ganglion. So the salivary nucleus in the medulla is important in the parasympathetic stimulation, and the superior cervical ganglion is important in the sympathetic stimulation. The transient increase of the secretion is due to the contraction of the myoepithelial cells due to the sympathetic stimulation. It causes vasoconstriction opposite to the vasodilation which is seen in the parasympathetic stimulation, which decreases the saliva formation and makes the secretion thick. There is also a reflex secretion, which the saliva is secreted reflexively by contact of the food with the mouth cavity. This is the unconditioned reflection, and this is by the birth in the humans. The next one is the secretion due to smell or thought of the food. This is the conditioned reflection, and the humans learn this by growing. Now let's talk about the applied physiology. The first one is the xerostomia. It is the decreased secretion of the saliva, which causes the dryness of the mouth and predisposes to the oral infections due to the loss of many enzymes which protect the oral cavity. The next one is the Sjogren syndrome. It is an autoimmune disorder which presents mainly with the xerostomia or the decreased secretion of saliva and xerophthalmia or the decreased tear formation in the eyes. It is common in women after the age of 40 years. So that's it for today, guys. I'll provide the notes of this video in my Telegram channel, which is Physiology at Once. I'll provide its link in the description. If you found this video useful and informative, please like, share and comment, and also don't forget to subscribe. Thank you.